A few weeks ago, we started with the idea of mitosis. And when we talked about mitosis, we said that mitosis made perfect copies of cells. So mitosis was very useful for growth in organisms. Mitosis was very useful for healing in organisms. But the other thing that mitosis is really good at is asexual reproduction. So asexual reproduction happens when the offsprings are only produced by one parent. So things like hydra, this green guy over here, that's a hydra, it's budding. Coral's bud, this is a single-celled euglena, it's just dividing. These things all reproduce asexually, which means the offspring are produced by only one parent. Each parent passes all of its genes to its offspring. Asexual reproduction happens through mitosis because, again, mitosis is the process of creating identical cells. So asexual reproduction definitely has some advantages. Number one, it's faster, and you can produce a large number of offspring. Most food poisoning bacteria can go from about one bacteria in the system to about 50,000 bacteria in your system in just a few hours, which is why you can eat, and then a few hours later you feel sick as a dog. Food poisoning, not fun. The reason why it's faster and the reason why we can produce so many more offspring is somewhat related to the fact that the parent does not have to find a mate. Okay, so it just speeds things up. However, there's some disadvantages too. All offspring are exactly alike. There is no variation inside the species. Therefore, anytime there's a change in the environment, that species is now going to be in danger of becoming extinct. That is, again, very problematic. In comes sexual reproduction. So sexual reproduction is the result of two parents. So a female gamete and a male gamete, a female parent and a male parent. And I do like to differentiate between gametes and parents because, like trees, for example, they do reproduce sexually, but they don't have like female and male parents. They have pollen, which is the male gamete for plants, and they have ova, which is the female gamete for plants, but there's not like a boy plant and a girl plant per se. All sexually reproducing organisms must have a male and a female. Again, these are male and female gametes. Now, in humans, we make sperm and females make eggs. But again, that is in humans. And obviously not humans don't represent every single type of organism that there is. So advantages and disadvantages to sexual reproduction. Disadvantages is this process is going to take a while because the parent has to find a mate. Also, fewer offspring are going to be produced. For example, it takes almost an entire year to make one human baby. It takes about 4 months to make, I think cats have about a 4-month gestation period. To make what, 4 or 5 cats, it takes 4 months. A bacteria can reproduce in the matter of hours, from 1 to thousands. And it just takes longer. So if it has all these disadvantages, why do it? Well, the biggest and most important benefit to sexual reproduction is the fact that all of your offspring are going to be genetically different from each other, okay? We create genetic diversity when we reproduce sexually. So, sexual reproduction involves gametes. Again, these are your sex cells. In humans, we call these eggs and sperm. And it involves fertilization. Fertilization happens when the sperm and the egg get together. When the sperm and the egg get together and there is a fertilization of the egg, we call that a zygote. So once the sperm and the egg get together and they share genetic material, we now have a zygote. A zygote, by definition, is a single cell where you have reunited a full set of chromosomal material. Again, half from mom, half from dad. This picture is what we call a karyotype. It is literally a picture of human DNA. Now, this human happens to be a female, and the reason I know that is because if I look right here, past chromosome set number 22, I see 2 chromosomes in the X position. For females, we will always have 2 X's. If it is a male, we will have an X and a Y chromosome. What we see here, ladies and gentlemen, are organisms that result from sexual reproduction. And the reason I know this is because I have 2 sets of chromosomes. I have 2 number 1s. I have 2 number 2s. I have 2 number 8s. I have 2 number 17s. I have 2 number 22s. I have 2 sets of chromosomes. Now, one of these sets came from the mom, and my other set came from my dad. All right, so half of my genetic material came from my mom. The other half of my genetic material came from dad. These matching sets, excuse me, these matching sets are what we call homologous chromosomes. Remember, homo, that prefix homo means same. So when I have a homologous chromosome, I have 2 copies of the same chromosome. One came from mom, one came from dad. What does it mean? Well, it means that I have 2 of the same chromosome that are about the same size, that carry the same genes, which code for the same trait. Now, there might be different expressions of that gene that create the trait. For example, like right here on set number 9, I got this nice dark area at the top of this chromosome. Let's pretend that dark area at the top of number 9, and the first one that I got from mom, what that codes for? Oh, I don't know. Blue eyes. And over here on that dark area on the top of number 9, the second number 9, this is the one that I got from dad, that top part is going to, let's say, code for brown eyes. Well, the top of these both code for the same trait. They have, therefore, the same genes, but we might have different expressions of that gene, how they show themselves in their phenotype. All right, so you are what we call a diploid organism. So what the heck does that mean? What that means is that in each cell in your body, you have two of each type of chromosome. Every cell of your body has 2 copies of all of its chromosomes. The symbol for diploid is 2N, where N represents the number of different chromosomes that an organism has. Now, humans are 2N because, again, we have 2 of each type of chromosome. Diploid cells contain 2 sets of chromosomes. We already know that. Now, remember, mitosis, I go from one diploid to 2 diploids. This is why mitosis works for growing. This is why mitosis works for healing. This is why mitosis works for asexual reproduction. I am simply developing more and more of my diploid cells. However, oh, what happened there? There we go. However, we are here to look at sexual reproduction, which means that we need to look at chromosomes and gametes. Now, eggs and sperm, which are the human gametes, the human sex cells, must have half of the number of chromosomes so that when we add them together, the zygote will have the correct proper number. So let's take a look at gametes of the human body. That would be egg. Each egg carries 23 chromosomes. I have a sperm cell carrying 23 chromosomes. When the sperm and the egg come together, they make a zygote, and that zygote has 46 chromosomes. And that's where that number comes from. It's not magical. I did not pull it out of the air. That's how I know. Again, I have 1 set plus 1 other set equals 2 sets. All right. Gametes are haploid cells. So this egg and sperm, these are haploid cells. Haploid cells sometimes are called 1N cells. Sometimes they're called sex cells because they only contain one of each type of chromosome. All right, so if I have cells that produce eggs and cells that produce sperm are called diploids, how on earth do I end up with an egg and a sperm that only is a 1N cell? Well, this is the process of meiosis, which we have been kind of talking about these last few days. Meiosis is a process of, we already said this, reduction division. So one more time, the process of mitosis, the whole process, the whole point is that we need to cut our total number of chromosomes in half while simultaneously dividing the cell, okay? So phases of mitosis, keeping in mind this happens only in sex cells. Again, for humans or animals, we're talking eggs and sperm. If we were in a tree, we would be talking pollen and ova. The purpose of meiosis is to reduce the number of chromosomes in half. Meiosis, unlike mitosis, like mitosis, is preceded by the replication of chromosomes. So if you guys remember interphase, that was the beginning of mitosis. Interphase is also the beginning of meiosis. Unlike mitosis, however, the replication is followed by two divisions. We go through meiosis 1, and then we go through meiosis 2. Thankfully, when we go through meiosis, we are still doing PMAT, which is exactly what we did before. Prophase, metaphase, anaphase, telophase. That doesn't change. We just do it twice. So this is what meiosis looks like. Meiosis 1 is up top. Technically, this one right here is part of meiosis 1, but whatever. And then we have meiosis 2. Right, so we start with interphase. Again, interphase is that resting phase. We're growing, we're replicating our DNA. We have then prophase 1, metaphase 1, anaphase 1, and telophase 1. Again, this is all the first round of cell divisions. Prophase, metaphase, anaphase, telophase, PMAT, it's still there, but this time we call it prophase 1, and metaphase 1, anaphase 1, telophase 1. Prophase 2, big surprise, metaphase 2. Anaphase 2, followed by telophase 2. All right, so I want to point out one of the differences right now between meiosis and mitosis. In meiosis, I start with one cell and I end with 4 cells. 1, 2, 3, 4 cells at the end. So one diploid cell becomes 4 haploid cells. All right, stages of meiosis. Interphase, just like before, interphase is where our chromosomes replicate. It's really, really similar to mitosis, and I end up with sister chromatids held together by a centromere. All right, for me, prophase 1 is probably one of the most important phases of meiosis. Just before, chromosomes short and thicken, not a big deal. But now each chromosome is going to pair up with its corresponding homologous chromosome, and that forms a tetrad. And a tetrad just means that there are 4 chromatids there. The most important part of prophase 1 is notice right here. See where these 2 chromosomes look like they're holding hands, and they look like they're holding hands down here. When you have a series of quote, quote, hand holding in prophase 1, what can happen is a process called crossing over. Literally, they are exchanging, those chromosomes are exchanging genetic information. This is why sexual reproduction causes so much genetic variation. This is one of the reasons why, even if 2 people have the same exact parents, they still look totally different. All right, metaphase 1, our tetrads line up at the center of the cell. So before we would just have one line going down the center, but now we seem to have like this double line going down the center. Anaphase 1, again, anaphase away, our tetrads break apart. So I have not broken apart my sister chromatids yet. All I am doing here is I am separating my homologous chromosomes to either side of the cell. The sister chromatids are still attached. All right, so now we are moving into telophase 1. And for telophase 1, just like before, they just separate into separate cells. So now I have 2 cells, but I am not done yet because I still have my sister chromatids attached. Can't have that. All right, so meiosis 1 results in 2 haploid daughter cells. However, like I said, the sister chromatids are still attached, so we need to keep moving. Prophase 2 is totally boring. Nothing exciting happens here. Literally, the sister chromatids just start to move towards the center. That's it. No crossing over, nothing special, nothing exciting. Prophase 2, totally boring. Metaphase 2. So our pair of sister chromatids now line up at the center. What I want to point out is that now it is a single file line down the center. In metaphase 1, I had a double line down the center. The tetrads were lined up, and now I just have a single chromosome down the center, a single solitary line. Anaphase 2. So now I finally am going to separate my sister chromatids at the centromere, okay? And those now, each individual single chromosomes are now going to move to opposite sides of the cell. Finally, in telophase 2, I end up with 4 brand new cells. So again, I started off with 1 diploid cell, and I'm ending with 4 brand new haploid cells. Now, oops, my bad. The most important thing to note here, ladies and gentlemen, is that not only are these 4 different from the parent cell, but these 4 are 100% different from each other. Each one of my haploid cells are 100% unique. That is where my genetic variation is coming from. So where do I gain genetic variation? Again, prophase 1 in the crossing over, metaphase 1 and metaphase 2, because how they separate is entirely kind of random. It's a random assortment in the metaphase. It doesn't say that, moms have to go one way, dads go the other. That's not how this works at all. So at the end of telophase 2, I have 4 100% unique individual haploid cells, which of course create genetic variation in this species. So the importance of meiosis. 1 cell that is diploid becomes 4 haploid cells. The chromosome number in the eggs and the sperm are cut in half, again, to ensure that the zygote will have the proper number of chromosomes, because all of this is being done so that we can eventually form a zygote, which is going to eventually develop into our offspring. Now, crossing over, literally, when this is happening in prophase 1, the homologous chromosomes line up next to each other, okay? So during prophase 1, each pair of chromatids lines up next to its homologous. So we get our homologous chromosomes lined up. The next thing that happens is that these pairing of homologous chromosomes produce tetrads. So again, I am counting 1 chromosome, 2 chromosomes, 3, and 4. I have 4 sister chromatids here. 2 chromosomes, 4 chromatids. I'm counting the chromatids, which make it the tetrad, 4. So what ends up happening? Oh. When they line up next to their homologous chromosomes, these chromosomes may twist around each other, and part of the red chromosome might break off into the blue and vice versa. They will have different sections attached to each other. Every time they do this, you get a different result. That is why sexual reproduction produces so much genetic variation. All right, this process is called crossing over because some of the red genetic material crossed over into the blue, some of the blue crossed over into the red. All right. Crossing over is an exchange of genetic information between two segments of homologous chromosomes during meiosis. And again, this is 100% related to genetic diversity. Because let's say, for example, if that original red chromosome was the one that you got from mom, well, now the mom chromosome has some dad genes on it and vice versa. All right? We are adding to our genetic diversity. All right, the result of the offspring will receive a new combination of genetic information, and this leads to a variation in the offspring. Variation leads to adaptation and change. These variations will cause some of the offspring to be better suited for particular environments. Remember, we talked about the changing of the environment and that being able to maybe allow you to survive, and sometimes you survive and thrive, and sometimes you don't. If they are better suited to the environment, it is more likely that they will survive to reproduce and pass on these favorable variation to their offspring. This is the heart of how evolution happens. The result of crossing over causes the offspring to be less suited to environment. It might not survive. And if the offspring doesn't survive, it might not reproduce. And that means it is not, these unfavorable genes are not likely to be passed off to the offspring. Okay? So, crossing over one more time, it leads to evolution. The whole reason why genetic diversity is important is because it leads to evolution, which we will be getting to in our next unit, but that's okay. We'll kind of put a pin in it for right now. All right, gamete formation. Meiosis, again, produces 4 haploid cells that are completely different. In the male, we produce in 4 sperm cells, and in the female, we produce 4 egg cells. All the cytoplasm and organelles go into 1 cell, and the other 3 are never functional. That is called, meiosis and mitosis. Mitosis, excuse me, occurs in all of the cells of the body except for your egg and your sperm. Meiosis only happens in the formation of egg and sperm cells. In meiosis, each diploid cell divides twice to produce a total of 4 cells. In mitosis, each cell, which is diploid, divides once to perform 2 cells. In meiosis, each of the 4 cells contain half the number of chromosomes as the parent cell. In mitosis, each of the new cells contain the same number of chromosomes as your original cell. In mitosis, the homologous pairs come together to form tetrads. In meiosis, the homologous pairs do come together to form tetrads, and while they are in tetrads, crossing over happens, and there will be no crossing over again in mitosis because there's no need for it. We're making identicals. Continuing the comparison. In meiosis, the 4 haploid cells contain different combinations of chromosomes from each other. In mitosis, the new cells contain identical